Ancient Mayan city discovered in the Mexican jungle. Using LiDAR, an aerial laser scanning technology, archaeologists have discovered the remains of an ancient Mayan city deep in the jungles of the Yucatan Peninsula. In Ocompton, which means stone column in the Mayan language. Researchers have found several pyramid-like structures over 15 meters high, a playing field and the stone columns that give the city its name. Researchers from Mexico's National Institute of Anthropology and History, INAH, have discovered the remains of an ancient Mayan city in the Balamcu Reserve on the Yucatan Peninsula. They named it Ocompton because of the numerous cylindrical stone columns scattered throughout the ancient settlement. In addition to the columns, pyramid-like structures more than 15 meters high, a playing field, plazas, terraces and structures arranged in concentric rings have been identified in the city. Researchers of the site also found pottery fragments that indicate that the site was inhabited between 600 and 800 AD, a period known as the Late Classic period. The Maya civilization stretched across what is now southern Mexico, Guatemala, Belize and parts of western Honduras. Most of these areas are covered with hard-to-reach jungle, which is why it was decided to conduct research using LiDAR, light detection and ranging. It is a measurement method based on irradiating the target with a laser and registering the reflection with sensors. Differences in the return time of the reflected light beam and the change in wavelength are used to create three-dimensional terrain models. Thanks to this breakthrough technology, which has taken a high place among the tools useful to archaeologists by storm, you can see the topography of the land without the top layer of vegetation. LiDAR penetrates the tree canopy and reflects the three-dimensional forms of archaeological features hidden beneath the vegetation. Researchers can digitally remove trees, plants, and even water from images, revealing hidden structures. LiDAR has already proven its usefulness. In 2018, with the help of this technology, in the Guatemalan jungle, the ruins of over 60,000 houses, palaces and other structures that had been hidden from the sight of modern man for centuries were discovered. Thanks to LiDAR, archaeologists also revealed the true size of the 40,000-year-old city of Angamuco, found a decade ago. Buildings and other structures. Thanks to this technology, the oldest and largest Mayan building, hundreds of ceremonial complexes from thousands of years ago, and the ruins of 11 cities and settlements belonging to an unknown culture were discovered. The research was aimed at broadening knowledge about a vast area practically unknown to archaeology. The 3,000 square kilometer site is located in the central part of the Mexican state of Campeche. The researchers focused on the northernmost part of the area. The research began in March. Aerial laser scanning by the University of Houston has helped archaeologists identify numerous structures in the uninhabited jungle. After the scientists LiDAR had collected the data, a trip to the area where the ruins had been found awaited. On site, researchers led by archaeologist Ivan Sprach of the Slovenian Academy of Sciences and Arts took a close look at the structures as seen from the air. According to them, the stone columns from which the place got its name could originally have served as entrances to rooms on the upper floors. The center, which consists of 3 large squares, is dominated by tall pyramid-shaped buildings surrounded by terraces. Between the vast squares, a playing field was found that may have been used for recreational and possibly religious purposes. There is also a complex consisting of various low and elongated structures arranged almost in concentric circles. The southern part of the city is connected by a causeway to the northern part, where the largest discovered structure is located. A huge rectangular square with sides measuring 80 meters by 10 meters was found there. There is a pyramid that rises 25 meters above the natural terrain. The biggest surprise was the site on a hill surrounded by extensive wetlands. Its monumental center covers more than 50 hectares and has various large buildings, including several pyramid-like structures over 15 meters high. The site served as an important center on a regional level, probably during the Classic period. The most common types of pottery we collected on the surface and in some test trenches date from the late Classic period. Analysis of samples of this material will provide us with more reliable data on the habitation of these areas, specifies Sprach. Greenhouse gas emissions are causing the stratosphere to shrink. The thickness of the stratosphere has decreased by 400 meters since the 1980s and will decrease by another kilometer by 2080. Scientists have determined greenhouse gas emissions are responsible for this. These changes may affect satellite operations, GPS navigation and radio communications. An international team of climate scientists has found evidence that man-made greenhouse gases have caused the stratosphere to contract. The findings, published in the journal Environmental Research Letters, show the profound impact humans have on the planet. The stratosphere extends from about 20 kilometers to 60 kilometers above the Earth's surface. Below that is the troposphere, a layer extending from the planet's surface to about 20 kilometers. The stratosphere, due to the ozone layer, plays an important role in absorbing harmful ultraviolet radiation. However, as recent measurements have shown, this layer is shrinking. Until now, the loss of stratospheric thickness has largely been attributed to the depletion of the ozone layer as a result of the widespread use of chlorofluorocarbons, CFCs, for many decades. However, The phasing out of CFCs has allowed the ozone layer to recover significantly over the past 20 years, alleviating concerns about permanent damage to the stratosphere. But new research reveals that the stratosphere continues to shrink even though CFC consumption has fallen and the ozone layer has recovered significantly, indicating that another factor must be driving this worrying trend. Previous studies have shown that the troposphere is thickening due to greenhouse gas emissions. This indicated a contraction of the stratosphere due to pressure from below, but the new study is the first to show this. In a new study, scientists sought to learn more about the impact of greenhouse gas emissions on the stratosphere. For this purpose, they obtained satellite data dating back to the 1980s. They added them to a computer model that also took into account chemical interactions in the atmosphere and the effects of the ozone layer. They also compared this data with climate models that take into account the increase in carbon dioxide emissions over this period. Such emissions cause the troposphere to heat up and expand, thus squeezing the stratosphere from below and compressing it. The models also showed that when carbon dioxide leaked into the stratosphere, it had a cooling effect. This drop in temperature forces the gases in this atmospheric layer to shrink in volume, leading to an even greater loss of the stratosphere. Calculations showed that since the 1980s, the stratosphere has shrunk by about 400 meters, which translates to about 1%. Its thickness. Further development of the models showed that the stratosphere would continue to thin as long as greenhouse gases were emitted into the atmosphere. Scientists suggest that the stratosphere could shrink by up to 1 kilometer over the next 60 years. The models also showed that changes in the ozone layer had little effect on stratospheric thinning. According to the scientists' data, the continuous shrinking of the stratosphere correlates well with the steadily increasing greenhouse gas emissions over the last 40 years, suggesting that our emissions are indeed responsible for this change. We found that the stratosphere has been shrinking by more than 100 meters per decade since 1980. We also proved that this is due to greenhouse gases, explained study author Juan Antonio Anel of the University of Vigo in Spain, in the most likely scenario of climate change. Our planet's stratosphere could lose 4% of its energy. Its vertical extent, or about 1.3 kilometers by 2080, he added. The researchers note that it is still unclear what impact the shrinking stratosphere may have on the planet, but indicate that it could affect satellite trajectories and the way radio waves propagate, ultimately affecting GPS navigation. Scientists have rejuvenated human skin cells by 30 years. Scientists have succeeded in turning back the age of human skin cells. The new method reverses the aging process by up to 30 years. Importantly, the cells do not lose their specialized functions. The authors of the study believe that it may be possible to do the same with other tissues in the body. This research is a huge step forward for regenerative medicine. Researchers at the Vabram Institute have developed a new method of skin cell rejuvenation. The technique allowed them to turn their clock back about 30 years, according to molecular measurements. This is much longer than previous methods. Importantly, the rejuvenated cells behaved like young cells in skin wound simulation experiments. The research, published in the journal eLife, although in its early stages, could have huge implications for regenerative medicine, especially if the method can be replicated with other cell types. As we age, the functions of our cells are limited and show signs of aging. This process can be tracked with tools such as epigenetic clocks, which measure the rate of change in the body to determine a person's biological age. Regenerative biology aims to repair, restore youth or replace older cells. One of the most important tools in regenerative biology is the ability to create induced stem cells, i.e. reprogramming older cells to a rudimentary embryo-like stage. These stem cells can then differentiate into any type of cell. This method was developed by a Japanese researcher, Nobel Prize winner, Shinya Yamanaka. In 2006, he discovered that differentiated cells could be reverted to an embryonic stem-like state by inducing the expression of 4 genes. This technique is now called Yamanaka transcription factors. Using Yamanaka's work, researchers at the Vabram Institute developed a technique that allowed human skin cells to jump back in time 30 years without losing their specialized function. Researchers were able to partially restore the vigor and function of older cells, as well as rejuvenate molecular measures of biological age. The Yamanaka technique allows cells with a specific function to be transformed into stem cells that can develop into any type. However, this means that the cell loses its specific identity and is difficult to induce to differentiate into the cell type from which it was withdrawn. But researchers at the Vabram Institute reprogrammed the cells to be biologically younger while retaining some of the functions that made them skin cells. The new method, based on a Nobel Prize-winning technique, is used to produce stem cells in a way that solves the problem of completely erasing the identity of the cells. All thanks to the modification of the Yamanaka technique. The new technique makes cells biologically younger while allowing them to regain their specialized cellular function. Yamanaka developed a technique to convert mature cells back into stem cells by exposing them to a mixture of chemicals for 50 days. However, after such treatment, scientists are unable to effectively induce stem cells to differentiate into mature cells of the same type again. To get around this problem, the authors of the new study exposed human skin cells to Yamanaka's transcription factors for just 13 days. Aborting the process about a quarter of the time prevented the cells from fully returning to an embryo-like state. causing them to regress in time only to an intermediate stage similar to the maturation phase. In this way, many age-related genetic markers were removed, and once the chemicals or transcription factors were removed, the cells were able to regain their specialized skin cell function again. Our understanding of aging at the molecular level has advanced over the past decade, giving rise to techniques that allow scientists to measure age-related biological changes in human cells, says biologist Diljit Gill of the Babram Institute in the UK. We were able to apply this in our experiment to determine the extent of reprogramming in our new method, he adds. Shinya Yamanaka was the first scientist to turn normal cells with a specific function into stem cells that have the special ability to grow into any type of cell. As already mentioned, the full reprogramming process of cells takes about 50 days. The new method, called transition to maturation reprogramming, exposes cells to Yamanaka transcription factors for only 13 days and allows the cell to retain its original identity and function. At this time, age-related changes are removed. In the experiments, the researchers gave the partially reprogrammed cells time to grow under normal conditions to see if their specific skin cell function would return. Later analysis showed that the cells regained markers characteristic of skin cells, which was confirmed by the observation of collagen production in the reprogrammed cells. After conducting the experiments, the researchers looked at a number of measures of cellular age, including the epigenetic clock, where chemical markers present indicate age. They also looked at the transcriptome, the set of mRNA molecules present at a particular point in time in a cell. These two measures showed that the reprogrammed cells matched a profile of cells 30 years younger compared to the reference datasets. The functioning of the reprogrammed cells also indicated rejuvenation. There are various elements in the skin, e.g. fibroblasts. They produce collagen, the main structural protein present in the skin, tendons and bones, which maintains the structural integrity of tissues and organs, is involved in the development or repair of tissues and organs, and in the process of wound healing. As the researchers noted, the rejuvenated fibroblasts produced more collagen proteins compared to control cells that did not undergo the reprogramming process. Fibroblasts move to areas that need repair. The researchers tested the partially rejuvenated cells by creating an artificial incision in a layer of cells in a petri dish and found that the fibroblasts they treated migrated to the wound faster than older cells. This is a promising sign that one day this research could be used to create cells that better heal wounds. With our approach, cells temporarily lose their identity as they enter maturation but, importantly, regain their initial somatic fate when reprogramming factors are withdrawn, the researchers write. Although the mechanism of this identity recovery is not fully understood, the authors speculate that it may rely on persistent epigenetic memory that is not completely erased during puberty. In addition, scientists have identified signs of rejuvenation in genes with functions other than fibroblasts, such as those associated with Alzheimer's disease and cataracts. Our results represent a major step forward in understanding cell reprogramming. We have proven that cells can be rejuvenated without losing their function, and that rejuvenation can restore some functions to old cells. The fact that we also observed a reversal of aging rates in disease-related genes is particularly promising for the future of this work. One of the next steps will be to try the techniques used here on other types of cells in the body and make sure the processes are completely safe before moving them from the lab to clinical trials.